Welcome to Making Waves.
Welcome to Making Waves.
Fresh ideas in freshwater science.
Fresh ideas in freshwater science and why they matter to you.
Making Waves.
Making Waves is brought to you.
Making Waves is brought to you with support from the Society for Freshwater Science.
Welcome to Making Waves. I'm your co-host, Erin Larson, and you're listening to part 1 of a 4-episode mini-series on education in freshwater science. In each episode, freshwater scientists will talk about an activity they've used in their class, ranging from activities that take just a few minutes to entire modules of a course. We hope these will help everyone from high school teachers through college instructors get new ideas to use in their classes. In this episode, I'll be talking with Whitney Beck, a PhD candidate at Colorado State University, about an experiment she does over multiple lab periods with an upper-level course. Welcome to the podcast, Whitney. It's great to have you with us today.
Thank you. I'm happy to be here.
Awesome. So I was wondering to start with if you could describe for us a little bit about the class context where you've done the activity that you'll be describing for us. Is it like an upper level class? What kind of type of university are you at, etc.?
Sure. So I'm at Colorado State University, which is a large public research institution, and I instruct a stream ecology and biology lab. These students are mostly juniors and seniors, and a lot of them are fish and wildlife majors, but we have some biology students mixed in as well. So it's about 25 students in the lab.
Awesome. And could you tell us a little bit about the activity that you do with these students? So how you prepare for it, and then also how much class time it ends up taking?
Yeah, so the activity I'd like to talk about today is a predator-prey size selectivity experiment. So this involves using crayfish, which are the predators, and snails, which are the prey, and kind of incubating them together in quart-sized containers to see if larger versus smaller crayfish consume more or less larger versus smaller snails. So it's a pretty basic type of experiment, but I think the students really enjoy the hands-on potential of it and coming up with hypotheses. They feel like they can understand ecologically what's going on and how it might translate to an aquatic ecosystem out in the field. So to prep for the activity, last year I actually went out and collected all of the crayfish and snails in October in Colorado,
which
was fairly difficult. I spent a lot of time with a kick net in the streams trying to find crayfish. I couldn't really see them to capture them, and I had just forgotten that colder weather means they're not as active. So that was challenging, and I think this year we might end up ordering crayfish. But then for the snails, I had to go out to my advisor, Dr. Poff's house. He has a water fixture in his yard, and we collected snails together and brought them back to the lab. So I think really the setup was just collecting these organisms, bringing them back to the lab, and then the students could work with them from there. But other than that, no specialized materials, which makes it really simple.
Nice. And so it sounds like this was sort of a half semester long activity, more or less, or how many class periods do you guys end up spending on this experiment?
Yeah, so we worked with the crayfish and snails for 3 different class periods. 2 of the class periods were the predator-prey experiment, where we set up the experiment 1 week. The students had to monitor snail deaths each day until the next class period, the second week. And then the 3rd week, we actually reused the crayfish and snails to do excretion experiments, where we put them in small vials and beakers and measured ammonium excretion and related that to organism size. So it was great to be able to use the organisms for multiple purposes.
Yeah, that's really cool. So it sounds like you'd sort of prepared the students for this activity beforehand, and then do they have to do data analysis afterwards? What types of, do they write up a lab report? What types of assessment do you use for this activity?
So to prepare for the activity, they first read a paper, Crowell 1990. It's about drying streams and predator-prey relationships and how size plays into all of that. And then after the activity, they use the data as the basis for a lab report. So we spend about half of a lab where they can work in groups, discussing the data, analyzing it in Excel, We used both regression and chi-square tests, so they get exposure to how you can use multiple techniques to analyze data sets, and then they turn in individual lab reports, so that's the assessment, and they're supposed to supplement the findings with background literature as well.
Awesome. So it sounds like a pretty complete view in some ways of scientific process from beginning to end for them.
Yeah, and I think one interesting thing about this kind of experiment is there is a lot that can go wrong, which helps them prepare for a career in research or post-grad careers. So we had crayfish that escaped from their quart-sized containers, and we found them on the floor. And we had sometimes it was hard to see if a snail was dead, like if a crayfish had pulled it out of its shell or if the snail was just really receded into its shell. Yeah, definitely some ambiguity in the process. So I think it was good for them to see mistakes and mishaps so they understand the need to replicate experiments really strongly. Yeah.
Very cool. And so it sounds like overall students reacted positively to this activity, but were there certain things that they really liked or they really didn't like that you noticed?
Yeah, so I think they, again, enjoyed having this hands-on activity, working with the crayfish and the snails, and they didn't seem to mind too much coming in outside of class to monitor snail deaths because it's such a random thing to do, and they're fish and wildlife students and often haven't thought about organisms outside of the fish world. So this is a whole new area of stream ecology for them. So just the novelty of it was interesting. I will say that they didn't, so when we used the crayfish and snails for the nitrogen excretion experiment, they did not enjoy filtering. And I thought that was funny because that's something we do so much in grad school. Like we have to filter the excretion water to actually run the water for ammonium. And I feel like I know a lot of stream ecologists and limnologists who spend hours of their grad school summers doing that. So I told them this is good grunt work to help prepare you for the hard labor later on.
Nice. And so it sounds like overall this is an activity that's worked pretty well with your stream ecology class the 2 times that you've done it so far. But have you made any changes between years, or would you change this in the future based on how things have gone in the past?
Yeah, one thing I would like to do is include the students a little bit more in the experimental design. So rather than just saying, here's what we're doing, here's what you have to work with, see if they have any ideas about how to better structure it, or if they want to include different treatments or response variables, like maybe they want to put a disc with algae in the tanks to see if snail herbivory changes in the presence of crayfish. That could be a really cool experiment too. So giving them some options and really giving them leeway to make it their own would be something that I want to incorporate in the future. And this semester I have a pretty small class. It's only 8 students. So I think that trying the bigger freedom and experimental design with a smaller class will be a good test, and hopefully we can incorporate that in future years if it works well.
Cool. Yeah, because in the past, have you been having the class analyze like the whole class data, or is everyone just working with their own subset of the data that they collected?
They analyze the whole class data.
Yeah.
But I do think with a smaller class, you could maybe split into 2 groups and have 2 different treatments and then have presentations about what you learned and share it with the rest of the class or something like that. So there's a little more flexibility.
Yeah, that's awesome. Yeah, you kind of end up sacrificing. We ran into something similar with the stream ecology class that I've helped teach at Cornell, where we do a leaf pack decomposition project, and we were trying to figure out how we negotiate letting students work with whole class data versus designing their own mini experiments where they might have fewer replicates to work with, which is definitely a trade-off for thinking about experimental design sometimes and data analysis and prioritizing one over the other.
Yeah, I had the same kind of experience with leaf pack decomposition data, where last year I did split students into groups and let them come up with their own treatments, but a lot of their replicates washed away and we didn't end up with a lot of data to work with in the individual groups, so we did end up combining everything into whole class data. So I understand that struggle as well. And I'm sure we all go through that at some point, trying to make those decisions.
Yeah, definite pros and cons. And so another question I had for you was thinking about, it sounds like you were teaching this for a pretty upper level majors class. And do you think you could, or would you be able to adapt this type of activity for like smaller, like, or more introductory classes? It sounds like you're already adapting it for a smaller class, but maybe for a larger class size than you've had before. Anything like that for folks who might be thinking about these types of class activities?
So I've thought a little bit about how this could be adapted for more of an introductory class, because the hypothesis-driven approach and analyzing the data in Excel and writing lab reports, those skills could be transferable to a freshman class or even a high school class or something like that. And what I think would be fun for students at an introductory level is to actually come out and help collect the organisms. If that was a chance for them to get out into the field and see the natural habitat of these organisms, they might really enjoy that. And then once we're in the lab, I think simplifying the experiment would be possible. So not varying crayfish and snail size, but just picking one, having like some monster crayfish in some tanks and then some tiny crayfish in other tanks. And they would have some intuition about the number of snails that each of those sizes might be able to eat. Another thing that could be really interesting for introductory classes is leaving the crayfish for a few days and then adding the snails so the students could actually see crayfish consuming snails potentially, because we didn't actually observe that a lot in these longer term experiments where the crayfish were fresh out of the stream and had leaves and other substrate in the tanks. So if we had some hungry crayfish and could actually see them in action, I think that would be really fun for intro level students.
Yeah, that sounds really cool. So Whitney, it sounds like you've had a lot of cool teaching opportunities so far during your PhD now that you're wrapping up. And so I was wondering if you had any advice especially for grad students who might be teaching in the aquatic sciences for the first time based on your experiences.
Yeah, so I will say that I was a little bit nervous at first because I am teaching mostly seniors who often have a vast wealth of knowledge, and I've tried to look at that more as an opportunity where I can be inquisitive with them and learn with them and from them as well. So providing structured time and activities, but also learning from them is something I've tried to do. So I'll give an example. We were on a field trip last week at a stream, and I just saw these logs that were covered in gelatinous masses, and I couldn't tell what they were. I'm a phycologist. I'm trained to study algae, and so my mind immediately went to cyanobacteria. But I brought the logs to my students, and they are fishery students, and so they said, oh, this is some type of egg, but I don't really know. And so we were kind of talking back and forth and wondering, and I brought the samples back to the lab. I took pictures of them, posted them on Twitter, and even respected professors were kind of arguing, what is this mass on the wood? Some people thought it was algae, some people thought it was snail eggs. And so I finally looked at it under the microscope, and it does look like some kind of gastropod eggs. Just having that freedom, though, to not be sure and to talk about it with students, and I think that makes a much more enjoyable experience for everyone.
Yeah, I completely agree. Thank you so much, Whitney, for taking the time to talk to us today. I really enjoyed it, and hopefully it's helpful for our listeners as well.
Yeah, thank you. It's been really fun, Erin.
You've been listening to the Making Waves podcast.
For more info,
please
visit us online
at the Society for Freshwater Science webpage.
Tune in next time
for another fresh idea in freshwater science.